Hello science fans and welcome to SHE-ensya. Our topic for today is part 2 of the principles of genetic transmission. In our previous video, we talked about how genes are selfish molecules that want to be passed on from one generation to the next in their unchanged form. This comes with the realization that genes are the first memes. What are memes after all? A humorous image, video, piece of text that is copied, often with slight variations, and spread rapidly by internet users. Barring the assertion on humor, isn't that what genes are? Discrete subunits providing instructions to make proteins that are copied, with minor alterations through mutations, that are spread from one generation to the next through sexual reproduction. And that is the primary means by which genes achieve immortality, by plants and animals having sex and creating offspring. But before fertilization and the creation of new life can occur, germ cells or gametes must first be created. As they are both processes of cell division, mitosis and meiosis have their similarities. The similarity begins with interphase, where growth stages are observed to produce the needed proteins to prepare for meiosis, as well as the creation of organelles to allow the cells to double its size. This will also be the portion where genetic content is doubled through the process of replication. The same terminologies are also used to refer to the different stages of meiosis, with parallels on the different events that are happening. Except that meiosis involves two cell division processes, and thus, we have meiosis 1 and meiosis 2. So let's dive into meiosis 1, which is also called reduction division. And it is during these stages when the cell is reduced from diploid to haploid. Diploid is the state of your mature cells where there are two copies of the chromosomes, while haploid is unique to germ cells or gametes and is a state where the cell only has one copy of each chromosome. In humans, our mature diploid cells have 46 chromosomes in total, 2 copies each of the 23 types of chromosomes. But our haploid cells, such as sperm cells or egg cells, will only have 23 chromosomes, 1 copy each of the 23 chromosome types. The first part of meiosis 1 includes prophase 1, which is similar to prophase of mitosis because it involves the degradation of the nuclear membrane and the activation of the organelles responsible for the creation of spindle fibers. But prophase 1 is a little bit more action-packed and can be subdivided into 5 substages. During leptotene, the chromosomes start to condense and the sister chromatids are united by the centromere. Zygotene is the second substage, and it is at this point when the homologous chromosomes become partially linked by the synaptonemal complex. And this synaptonemal complex becomes important during the third stage, pachytene, where crossing over or an exchange of genetic content between homologous chromosomes occur. That's the most interesting thing about meiosis. It has the capacity to create chromosomes that have never existed before by swapping pieces of DNA from one chromosome to another. And this is when the selfish gene compromises, because evolution has shown that it can survive better and achieve immortality by working in tandem with other genes and promoting the survival of the vessel that they ride on. That's basically us, humans, and other plants and animals. Crossing over ends during the diplotene substage. The chromosomes, however, remain linked through visible structures called chiasma. And prophase 1 ends with diakinesis. And here, the chromosomes spread out where portions not linked by the chiasmata move apart from each other. As the cell transitions from prophase 1 to metaphase 1, the spindle fibers attach to the kinetochore in the centromere of the chromosomes, and the homologous chromosomes align at the equatorial plate of the cell. At this point, I would like to highlight the difference between metaphase of mitosis and metaphase 1 of meiosis. In metaphase of mitosis, single chromosomes align at the equatorial plate, whereas in metaphase 1 of meiosis, homologous chromosomes side by side and connected by chiasmata are the ones that align at the equatorial plate. When anaphase 1 proceeds, the chiasmata between the homologous chromosomes break, and the homologous chromosomes are separated. Again, in contrast with mitosis, anaphase will result to the separation of sister chromatids. But anaphase 1 of meiosis will result to the separation of homologous chromosomes. It is also notable that it is still cohesin that maintains the bind between the homologous chromosomes in anaphase 1, and their separation is facilitated by separin and the shortening of the spindle fibers. In telophase 1, cytokinesis occurs but no complete reformation of the nucleus happens. The chromosomes also only partially relax and no true uncoiling happens. So, at the end of telophase 1, each of the two daughter cells that are created will only have one copy of each chromosome type. And at this point, they are now considered haploid. Meiosis 2 occurs with no further replication of the genetic content. And because of this, it is called the equational division. And this is because it evenly divides the two already existing haploid cells and transforms them into 4 haploid daughter cells. Prophase 2 is considered to be the fastest stage of meiosis. This is because at this point, the barely reformed nuclear membrane starts to disintegrate once more, and the barely relaxed chromosomes recondense, and finally, the spindle fibers reattach to the centromeres of these chromosomes. In metaphase 2, the chromosomes align at the two daughter cells' equatorial plates, very similar to metaphase of mitosis. And when anaphase proceeds, the two sister chromatids finally separate. At this point, I would like to call back to the crossing over that occurred in prophase 1. This means that the sister chromatids no longer have exactly the same information. So, when they become separated, you will end up with chromatids that have distinct new information within them. And thus, at telophase, the cytokinesis event and the reformation of the nucleus would result to the formation of 4 distinctly different haploid cells. By the way, at this point, the chromosomes finally relax to chromatin. The germ cells will not rest at this point. They will continue to encounter conformational changes that will prepare them for fertilization. And it is during fertilization events, particularly when a new organism is formed, that our selfish genes get transferred from one generation to the next. And it is at this point where genes express their selfishness. If evolution was to progress, we would be perfected over time. But as you can see, genetic disorders continue to be passed on from one generation to the next. Inclusive fitness exists in such that evolutionary value of a characteristic is weighed not simply on whether it helps that individual to survive, but whether it makes it more likely that their offspring will survive. It doesn't matter if a combination of genes is harmful, as long as there are enough genes present that will allow the survival of the offspring of the organism. Even genes that harm the individual that carries it will want to be passed on to the next generation. And though the gene is the basic unit of selfishness, I hope it doesn't hinder us from behaving in such that it will benefit society in general. I hope you were able to learn something in this short video about the processes involved in genetic transmission. I hope you can give this video a thumbs up and also subscribe to our channel. If you have any comments, questions, or suggestions, please don't hesitate to message me, your resident Filipino scientist, in the comments section below. Thank you very much, and see you around!